Millions of genetically modified mosquitoes, killer bees that escaped a lab and spread across 2 continents, hybrid snakes, genetically altered crops, and more. These are genetic experiments that created something scientists couldn't control. Back in 2013, a British company called Oxitec released millions of genetically modified male mosquitoes into a city in Brazil called Jacobina. These males didn't bite people, so that wasn't a worry. The idea was that they carried a gene that would stop most of their babies from surviving, so if enough of them tried mating with wild females, the mosquito population would shrink. That would help slow diseases like dengue, Zika, and yellow fever. And it worked, at least at first. Mosquito numbers did drop by a lot, but scientists kept checking on the population for years afterward. In 2019, a study came out with a bit of a surprise. Turned out some of the released mosquitoes had successfully bred with wild ones, so pieces of their DNA had stuck around in the population and obviously was not the intended goal here. Now, Florida already has enough strange stuff going on without adding giant snakes into the mix, but that's exactly what happened. Burmese pythons made their way into the state after being sold as exotic pets, and when they started escaping into the wild, it kind of took over. By the late 80s, they built up a real population across parts of Florida and started causing serious damage to wildlife. These snakes can get massive, up to 23 feet long and close to 200 pounds. Eat pretty much anything they can overpower, which throws the whole ecosystem out of balance. Well, then in 2018, researchers digging into the population found something even more unexpected. Out of 426 snakes they tested, 13 were hybrids. Not just Burmese pythons anymore, mixes with the Indian python too. The Indian python is smaller and quicker, so when it mixes with the Burmese python, you get a pretty deadly combination. A snake that's big and strong, but also very agile. Basically a tougher, more adaptable version of an already serious invasive predator. When CRISPR showed up around 2012, it was pretty exciting news. Being able to edit DNA, cut out what you don't want, swap in the good part, sounds pretty ideal, but as scientists started using it, they started noticing things the early tests had missed. In 2018 and 2019, several research teams found that CRISPR wasn't always making those small, neat edits that everyone had hoped for. Sometimes it deleted thousands or even millions of DNA letters instead of just a few. Other times, whole sections of chromosomes got rearranged or flipped, and the unsettling part was that a lot of these mistakes didn't show up in the usual tests scientists used to check their work. Everything looked normal until people dug deeper. Didn't mean CRISPR was unsafe across the board. It's shown real promise treating certain diseases, but it was clearly a lot more complicated than they thought before. Best way I understand it, instead of correcting, say a typo, it's more like ripping a page out, taping it back in, and only later realizing a whole paragraph went missing. Labs started building better tools to scan edited DNA before using it in treatments, and scientists began looking for ways to make CRISPR more precise with less side effects though. Fish farmers have wanted faster growing salmon for decades. A normal Atlantic salmon takes about 3 years to reach market size. A company called AquaBounty figured out how to cut that time almost in half by adding a growth hormone gene from Chinook salmon plus DNA from a fish called the ocean pout. The result was the Aqua Advantage salmon, the first genetically engineered animal ever approved for people to eat in the US. Yum. Now, aside from how off-putting it is to think about eating lab fish, what worried scientists more was what might happen if these salmon ever got loose. Since they grow faster and mature sooner, a big worry is them outcompeting with wild salmon, just disrupting the natural population. To prevent that, AquaBounty built in some safeguards. The fish are raised on land. Instead of in ocean pens, they're kept behind physical barriers, and almost all of them are sterile females, so breeding, even if one escaped, is impossible. The FDA decided the odds of these fish ever establishing themselves in the wild was extremely low, since every one of these safeguards would have to fail at once. Still, environmental groups kept pointing out there's no foolproof way to prevent anything. Farmed fish have escaped before. In the 90s, genetically modified canola spread across farms all over Canada. These plants were built to survive certain herbicides, so farmers could spray their fields to kill weeds without hurting the crop. At first it seemed like a perfect fix, but within a few years, something strange was happening outside the fields. Wild plants and weeds growing along roadsides started showing the same herbicide-resistant genes. Canola pollen travels through wind and gets carried by insects, so it spreads pretty easily to other plants. So now, typical weed killers were useless against what were basically superweeds, which is what farmers started calling them. Studies in Canada confirmed that this was pretty much permanent too. Once these traits got into the wild, there was no realistic way to pull them back out, so that's terrific news. Next, we're going to talk about gene drivers. A gene drive is a way of genetically engineering an organism so that a specific gene gets passed to almost all of its offspring instead of only about half. Normally, a gene has a 50-50 shot of getting passed down. A gene drive breaks that rule and just forces itself into almost every offspring. So in theory, scientists could use this to spread a positive trait through an entire population, like making insects incapable of carrying disease. That sounds great. So in a lab experiment with fruit flies, researchers used CRISPR-based gene drives to pass a genetic change down through several generations, and at first it worked great. The trait spread through the test population way faster than you'd get in nature, but as the experiments kept going, things started falling apart. Some flies developed mutations right at the spot CRISPR was supposed to cut, which basically made the tool stop working on them. Those flies passed resistance down to their offspring instead of a gene drive, and that resistance started spreading too, which slowed the whole thing down, and eventually it just stopped working. Point is, evolution is a lot more powerful than what we can cook up in a lab, for better or for worse. Rice is one of the most studied crops in genetic engineering, since it feeds billions of people. Scientists have used CRISPR to try and improve things like drought resistance, yield, disease protection, usually through tightly controlled field trials. But even in these controlled settings, researchers have run into problems, mainly one: genes moving beyond the test areas. Rice pollen travels through wind and water, and it can get a lot further than you'd expect. In some of these field studies, gene flow from experimental rice turned up in nearby plants that were never supposed to be edited. Even a little cross-pollination can introduce engineered traits into nearby crops. Once that happens, separating the modified ones from the rest gets tough, especially in places where rice covers huge areas. Researchers also found that wind, insects, and how closely plants are spaced all affect how far pollen travels, which makes containment a lot harder than just fencing off a test field. And here's a perfect example. In 2013, a farmer in Oregon found genetically modified wheat growing in a field where it absolutely was not supposed to be. Weird part was that this wasn't even an approved crop. An American agricultural biotechnology corporation called Monsanto had developed the wheat years earlier to survive glyphosate, a popular weed killer, but had only ever been used in field trials. It was never approved for commercial planting anywhere. So naturally, people wanted to know how it got there. The USDA launched an investigation to figure out what happened. The original field trials had taken place in the early 2000s across states like Oregon and Washington. Once they were finished, every plant was supposed to be removed, but wheat produces an enormous number of seeds, and it doesn't take much for a few to get left behind. The USDA found no evidence that wheat had made its way into the food supply in any meaningful amount or posed a health risk, so they say, but that wasn't really the point. Somehow this experimental wheat had shown up again more than a decade later. The discovery was scary enough. Japan temporarily paused some US wheat imports. Just another reminder that once some lab-engineered thing gets out into the wild, making sure every trace of it is gone, it's a lot harder than it sounds. This story starts in the 50s when scientists in Brazil tried to improve honey production by crossbreeding European honeybees with African honeybees. The goal was to create a bee that produced more honey and handled tropical climates better. Well, in 1957, some of the experimental queen bees escaped from a research facility in Sao Paulo. Once they were loose, they started mating with wild bees, and the hybrid population that formed spread way faster than anyone expected. These Africanized bees, as they were called, acted differently than typical European honeybees as well. They defended their hives a lot more aggressively, which is how they ended up with the nickname killer bees. Over the next few decades, these hybrid bees spread through South America, Central America, and eventually into the southern United States. But they didn't just stay the same as the original lab bees. Once they escaped, they kept breeding with wild populations and continued evolving on their own. In other words, scientists completely lost control of where those genes would end up. Today, Africanized honeybees are still found across parts of the Americas. They're no more venomous than regular honeybees, but again, they are more aggressive. That's been the video. I'll catch you, yes, you specifically, in the next one.